everyone, welcome to my VELS Phytopharmacy channel. Today I am going to take present revealing bioactive compounds, a modern systematic approach to phytochemical analysis part 2. This is my content of the slide. Conventional method of extraction, maceration, percolation, distillation and identification, followed by non-conventional method, modern techniques used for the extraction, qualitative analysis, quantitative estimation of phytocompound, chromatography techniques, in that one characterization of bioactive compound by spectral method of analysis and conclusion. Coming to the conventional method of analysis, it is a maceration involves the choking of the plant material in a solvent to extract bioactive compound. Typically, it is carried out at a room temperature in a cold condition for gradual efficient solvent penetration. It is the simplest and easiest method. In maceration, the whole or coarsely plant powder material is kept in contact with the solvent in a stoppered container for a definite period of time with frequent agitation until the solute particles completely extracted or dissolved. This method is best used in the case of the thermolabile substances. Coming to this oxalate, it is a hot method of extraction. It is a continuous process. Here it is less importance thermolabile substance because heat is used. It is a assembled apparatus consists of the round bottom flask, extractor and condenser. Place the solid sample which containing bioactive compounds in a thimble which is made up of either Whatman filter paper or muslin cloth. Insert the sample into this oxalate extractor. Heat the solvent in the flask to evaporate the solvent. The evaporated solvent passes through the long tube, enter into the condenser where it was cooled by the circulated water. Then it reaches into the extractor where the extraction was automatically carried out. When the extracted solvent layer reaches the siphon arm, it automatically drains out into the round water flask, back into the round water flask. This cycle was repeated until a concentrated extract is obtained for an overall period of time. This process continues until the extraction is completed. It was ensured by the presence of the chemical test, or you have to do the chemical test, ensuring efficient recovery of the bioactive compounds. Next, hydrodistillation. It is mostly applicable for the isolation of the volatile compounds, mostly terpenoid volatile oils from the plant material. Here the organic solvents are not much used. Only steam or water is used as a solvent. There are three types of distillation: hydrodistillation, water distillation, steam distillation and direct steam distillation. Here the plant material which is going to be extracted was packed in the extractor still. Water was added to bring to boil and injected steam can be applied from the steam boiler. Heat releases the bioactive compound, thereby the both volatile compound also and water also vaporize. The vapor passes into the tube into the condenser where both can be condensed to get the essential oil which can be collected in the receiver. Cool the vapor mixture, condensing it, separate the oil of the bioactive compounds from the bioactive compound and water in a separator. It is the most applicable for the simple and easiest technique, time-less process also. Then modern methods of extraction. Here the modern methods of extraction, otherwise known as non-conventional method of extraction. Here it is most focused on for the following merit: efficient extraction, speed is there, less time consuming substance, more sustainability. Moving beyond the traditional maceration, percolation, decoction, infusion method, these are the very important techniques. Supercritical fluid extraction, using supercritical fluid, mostly the inert gas carbon dioxide used. Microwave assistant extraction, ultrasound assistant extraction, pressurized extraction can be used. Here eco-friendly solvents like water or nano solvents can be used to quickly release the bioactive compound and preserve the quality of the extract also, and produces the some efficient extraction. These are some of the modern techniques used for the extraction of the phytocompounds. Supercritical fluid extraction here uses supercritical carbon dioxide for efficient extraction. Enzymes break down the cell's wall for extraction, enzyme assisted. In microwave, high frequency radiation was applied. Rapid extraction, heat is used. That by using heat, penetrate the cell wall, thereby the bioactive compounds can be diffused. Pulse electric field, here somewhat electric field pulsalized electric pulses can be used for the preliminary extraction of the phytocompound. Ultrasound assistant extraction, here ultrasound radiation frequency was applied, which produces some cavitation of the bubbles in the sample. Thereby it causes the penetration of the plant material, thereby extraction of the bioactive compounds occurs. Pressurized efficient extraction that can be also used for the utilize the high pressure for efficient solvent penetration and efficient extraction of the sample. Supercritical fluid extraction. It is an innovative separation of techniques. Here we can use for the isolation of the alkaloid, decaffeination, terpenoids. Here any substance or any gas or any liquid can act as a supercritical fluid under critical temperature, critical pressure. By using when it becomes a supercritical fluid, it have a more diffusion property, more penetration property. Due to that quality, it can be utilized as a supercritical fluid. Mostly carbon dioxide can be used as here because carbon dioxide is an inert gas, chemically inert. It is easily available. Recovery is easy and efficient extraction less expensive. To efficiently extract, offering benefits such as non-toxic solvents to selective separation for the various applications in the industry. This is assembled, this is apparatus. Here it is called as a still, where the plant material, coarsely powdered plant material was taken. Then the carbon dioxide from the still gas generator, it was supplied. The pressure can be adjusted. Due to pressure valve adjusted, the carbon dioxide became a liquid. Due to liquid, carbon dioxide penetration will be more efficient, more diffusion property, more penetration property to disturb the cell wall, thereby it increases the extraction process. So the caffeine can be separated along with the carbon dioxide liquid from the coffee beans. Both can be collected in the separator, where the pressure is adjusted by using pressure valve. The degassing can be done, so that the distillation unit caffeine can be collected. The recovered carbon dioxide again back into the still. Like that we can use carbon dioxide as a simple one. Mostly this supercritical fluid extraction is a less time consuming process, more efficient, eco-friendly. There is no need of the solvent. We can use any gas, inert gas can be act as a supercritical fluid. Coming to the microwave assistant extraction, here the pressed plant material was collected from the plant, dried under the shade, then it was coarsely powdered. The coarsely plant material mixed with the solvent. The solvent was kept in the microwave assistant extraction, microwave device. Here high frequency microwave at a particular frequency level or particular temperature can be applied. So what happened that microwave radiation will penetrate into the powder material, which causes the movement of the particle. Due to movement of the particle in the sample, heat is produced. With the help of the heat, disturb the cell wall of the plant cell, then more extraction can be occur. But here the more important thing we have to observe, the frequency we selected for microwave, the temperature we selected for that one should be controlled one. It should be optimized one. Otherwise, if excess heat, excess frequency of wavelength is used, there is a degradation of the plant material occurs. After that one, the extract was collected, which was filtered and concentrated. Then it can be used for the qualitative analysis. It is ultrasound assistant techniques. Here, the plant material was kept in the solvent, powdered plant material. Then particular frequency sound waves can be applied into the plant material. So these frequency sound waves will causes the bubbles in the sample. There is a cavitation of the bubbles occurs, bubble growth. The bubble causes the disturb the cell wall. Due to cell wall disruption inside the cell wall, the phytochemicals can be released. So the phytochemicals, selective phytochemicals can be used. So high temperature, high pressure radicals can be used here. So that can be causes the breakdown of the cell wall. Thereby the compounds are released. Here there is a limitations are there. We can use optimized ultrasound frequency waves condition. At the same time, if you use excess of the ultrasound radiation, there is a chances for the degradation of bioactive compounds. Thereby the extraction of the natural compounds cannot occur. So this is mostly used for the isolation of the flavonoids, polyphenols and carotenoids. Coming to the enzyme assistant extraction, most of the plant cell wall is made up of made up of the cell wall constituents like cellulose, pectin, hemicellulose, and some glycosides. So by using natural enzymes like cellulase, pectinase, beta glucosidase, or any hemicellulase enzyme, proteinase enzyme, we can assist the extraction process. Those processes which are done with the help of enzyme is called enzyme assistant extraction. By using this technique, we can isolate the glycosides, polypeptide, proteins from the plant cell. So here the enzymes are added along with the plant material in the extraction with the solvent. So thereby the enzymes causes the disturb the cell wall efficiently, enhancing the extraction of the valuable compounds while maintaining the mild processing conditions. Coming to the qualitative analysis, in any phytochemical analysis after the extraction, whatever may be the method, the nature of the bioactive compounds was identified, because all the extract whatever may be, it contain different type of compounds, because the plant is a multi-component system. So we have to do the simple chemical test in the laboratory level. By using laboratory reagent, we can identify whether the extract contain alkaloids, flavonoids, or tannin. Sometimes the one extract contain more than 3 or 4 type of compound. That was evaluated by simple chemical test in the laboratory, qualitative chemical analysis. So terpenoids can be done, carbohydrates, proteins, glycosides, like that one. Then after identification of the nature of the compound, we can determine the how much quantity of the particular compound present in the particular extract. That is called quantitative analysis. That can be done by UV spectral analysis or visible analysis or even HPLC method. So here a particular wavelength was applied into the sample. Then it was compared with the standard curve and standard by interpolation method. These are the some of the references I quoted here for the identification of phenolic compound and total flavonoid content. So qualitative quantitative analysis of phytocompounds can be done either by gas chromatography method coupled with the mass spectroscopy, or by high performance liquid chromatography or high performance thin layer chromatography. So in GCMS can be applied to solid, liquid, gaseous sample. First the samples are converted into gaseous state, then analysis is carried out on the basis of the mass to charge ratio by using MS technique. High performance liquid chromatography, here a pressurized solvent can be used to produce the precise separation and precise analysis method. High performance thin layer chromatography is applicable for the separation, detection, qualitative, quantitative analysis of the phytocompounds. Gas chromatography is applicable for volatile compounds. In this method, species are distributed between the gas and the liquid phase. The gas phase is flowing on the phase in stationary. When the sample molecules are in liquid phase may be stationary. The rate of migration depends upon how much of chemical species distributed into the liquid phase. In gas chromatography, the gas can be act as a mobile phase, whereas the column was packed with some solid material. So here the separation takes place based upon the affinity coefficient between the stationary phase and mobile phase. The species which distributed itself 100% in the stationary phase will not migrate. They are retained, that produce, they are eluted from the column, they are detected in the detector and signal. If a sample have more affinity towards the mobile phase, they can be easily passed out along with the solvent, not a good separation. The gas chromatography gives us the total vapor pressure value that is most widely used for the separation of the compounds. Coming to the high performance liquid chromatography, as the name itself here, pressurized solvent can be used. It separates the compounds on the basis of the interaction with the solid particles of a tightly packed column and the solvent of the mobile phase used. Here high pressure used, about 400 bars can be used. So here HPLC is useful for the compound which are non-volatile in nature, which cannot be vaporized. So in that condition, this technique is most commonly used. So here the compounds decomposes under high temperature. Those compounds, they are not vaporized. Those compounds, they are very sensitive to temperature. These compounds can be isolated by using this technology. High HPLC provides both qualitative purpose also and quantitative purpose also. Coming to the high performance thin layer chromatography, it is a advanced technique of the TLC, whereas TLC is a conventional method. We cannot use in TLC with the particle layer, the adsorbent thickness layer will be only 12 to 12 micron about, whereas in high performance thin layer chromatography they are pre-coated with the sorbent. Their particle size about 5 to 7 microns and a layer of thickness 150 to 200 microns. The reduction in thickness of the particle size results in the increasing efficient extraction along with the nature of the separation. So HPLC also used both for qualitative and quantitative analysis. High performance thin layer chromatography is a modified version of the thin layer chromatography technique. Here the sample can be applied by using automated, semi-automated manual injector application. Then the layer can be done by the development also done in the outside. Nowadays, a modern super techniques are also used. Characterization of the bioactive compound after chromatography isolation over, the compounds are characterized by using spectral method of analysis. So modern phytochemical analysis, these are the some of the modern phytochemical techniques are used for the molecular identification, structure elucidation, structure characterization, FTIR and other things are here. Mostly there is a single technique is not at all possible for characterization of the compound and structure identification of the phytocompound. So always you have to combine technique, either GCMS or LCMS or HPLC with the FTIR, like that only we can use. It enables the rapid screening and comprehensive analysis of the complex plant extract for the production of for the isolation of the lead molecules and for quality controlled process. So this is another technique, UHPLC coupled with the MS. So this is ultra high performance liquid chromatography, which is coupled with the mass spectroscopy, provides a precise mass measurement of the identification of the compounds like flavonoids and phenolics. GCMS is also ideal for the isolation of the volatile compounds like terpenoids, essential oils and fatty acids. So those compounds, they are volatile in nature, they are easily degraded by heat, they can be usually used for here. FTIR spectroscopy, it is a Fourier transfer infrared spectroscopy which identify the specific functional group like carbonyl group, conjugated system, aromatic diene and aliphatic ion can be detected infrared light absorption. So here the sample absorb the infrared light and providing a quick fingerprint within that stipulated time. Ultraviolet visible spectroscopy, this is another technique which can be used for qualitative quantitative analysis, and also it is used for the quantitative estimation of the phytocompound. Here the sample was observed either visible or ultraviolet at particular wavelength. Therefore the molecules are excited, so they produces the absorption can be done by the here in FTIR the molecules the spectrum produces only the functional group. The absorption takes place by the chemical bond. Here the absorption takes by conjugated system. So this is used for the chromophores and conjugated systems, anything used for quantifying certain compound classes like phenols and other things. Coming to the NMR, nuclear magnetic resonance helps to determine the detailed molecular structure and stereochemistry by analyzing atomic nuclei in a magnetic field, often used along with the LC, that is liquid column chromatography, or GC. Here the sample magnetic radiation was applied into the sample. There is an interaction between the nuclei of the sample with the magnetic field. Thereby it produces there is a internal structure can be disturbed. Thereby each and every atom produces a different signal, different absorption. Based upon the absorption of the interaction with the nuclei and the magnetic field, it produces a different signal. By using the this one, we can easily identify the sample. Coming to the capillary electrophoresis mass spectroscopy, here it is a capillary electrophoresis. It is here it is a separation of the ionic compound can be done under the influence of the electric field. There is anode and the high voltage can be applied. So if that when the compounds are isolated, which is coupled with the mass spectroscopy. In mass spectroscopy, it is a the compounds can be identified based upon the mass charge ratio, offering a high efficiency separation extraction. HPTLC, it is a advanced technology of the TLC method. Here better resolution and quantification for early stages of screening. The principle of HPTLC is same as that of the TLC, either differential partitioning or adsorption, difference in the partition coefficient or difference in the adsorption of the compounds between a stationary phase and a mobile phase. HPTLC plates are so sorbent particles that are much smaller, typically 5 to 6 micrometer, high narrow particle size are there, distribution compared to the traditional TLC method where the particle size are about 12 micrometer. Higher affinity for the stationary phase move slower, whereas higher affinity towards the mobile phases cannot be moved. They are move faster, so they cannot be separated. So based upon the affinity towards the mobile phase and towards the mobile stationary phase, the compounds get eluted or it can be retained. By using whatever may be the analysis, phytochemical analysis, the phytochemical analysis are mostly used for the drug discovery process, that is identification of the bioactive compounds from the herbal medicine for the generation of the new medicine. And also it produces ensuring the consistency and purity of the herbal matter, thereby the quality control of the herbal products as well as herbal supplement nutraceuticals can be determined. Metabolic studies also carried out in plants under their biochemical pathway. Apart from that one, they are used for in forensic medicine, in toxicology, in pharmacology, and also determination of the pesticide content, in quality control, in standardization of the compound. Any herbal systematic analysis, this spectral chemical analysis can be used. Coming to the conclusion of our slide. So plants are the richest source of the bioactive compounds. Bioactive compounds have less side effect and more targeted therapeutical potential because of its synergy properties and chemical identity. Isolation and identification of its identity is a great challenge for researchers. Still, so far more than 500 bioactive compounds were used in market for various ailments. So this presentation covered the modern systematic approach from basic qualitative testing to advanced green extraction like supercritical electric pressurized extraction, microwave extraction techniques, and highly resolution analytical technique which produces the combined techniques to identify the specific interesting compounds from that one. Thank you. These are my references. Thank you.